Happy 14 Juillet to everybody. As we discuss a brief history of poliomyelitis, it's really a pleasure and a privilege to have our colleague, Professor Vincent Racaniello, here. He's made many distinguished contributions to the understanding of viruses, including making the first DNA copy of an RNA virus and demonstrating, in this case polio, demonstrating that it's infectious, which led to many new avenues in virology and in the capability of changing viruses and studying viruses in many new ways. Professor Racaniello is the Higgins Professor of Microbiology and Immunology at Columbia at the College of Physicians and Surgeons, and also teaches a virology course here on the Morningside campus. So we're delighted to welcome him back to discuss a brief history of poliomyelitis. Thank you.
Thank you, Stephen. Good to be back. So today I'm going to tell you a little bit about the history of polio. I've always felt that I've been lucky to have studied polio because this is a virus with a very rich history, and it has given me appreciation for the necessity to have to talk into the microphone. Yeah. There's no mobile mic.
There is.
Yeah.
I do like to wander. I like to go up to people and ask them questions.
No.
Thank you very much. So I've been lucky with polio to have a great history. I'm going to try and give you a sense of that today. But let's start first by, can anyone tell me who I stole this title from? Anyone know?
Stephen Hawking.
Stephen Hawking. Who said that? You? Very good. I don't have a prize for you, but that's right. So about 15 years ago, I read A Brief History of Time, and I was impressed that a scientist, a very good scientist, could convey what they're doing to the public. And that, at that time, inspired me to do some of the things that I'm doing today, reaching out to the public and trying to teach them about viruses. And I'll tell you a bit about that later. What I would like to do today is to give you an overview of the history of this virus as a paralytic agent, but we're going to mix in some biology because I am a research scientist. I've worked in the lab on polio my whole career, and I want to give you some of that because I think it's important to understand the history of this disease to understand the agent as well. But first, we'll define poliomyelitis, the disease caused by this virus. The word comes from the combination of two Greek roots, polio for gray, myelon for matter, poliomyelitis, anitis, of course, the Latin meaning inflammation. And here is a description of the disease from a 1959 textbook of medicine. This is no longer in textbooks of medicine, of course. A common acute viral disease characterized clinically by a brief febrile illness with sore throat, headache, and vomiting, often with stiffness of the neck and back. In many cases, a lower neuron paralysis develops in the early days of illness. So we will actually deconstruct that statement completely, and we'll try to explain how the virus does this. As far as we can tell, this disease has been around for a long time. We don't have any clinical isolates from the 18th Egyptian dynasty, but we have this carving or stele which shows this priest with a dropped foot, which is very typical of polio. Maybe it's not polio, but a number of individuals have suspected that it might be, and this dates from 1580 to 1350 BC. Throughout history from this point on, there are sporadic descriptions of various diseases which may or may not have been polio, so we don't have a coherent record, but I suspect that the virus has been around for thousands of years, in humans at least. The first very good clinical account of this disease was made by this London pediatrician Michael Underwood in 1789 in a book called Treatise on Diseases of Children. He is clearly describing poliomyelitis in this book. And from the Egyptian stele up until this point, as we said, we have sporadic descriptions of what could be this disease. So I suspect it was around for a long time, but it was quite rare and did not occur in epidemics, as we will see later. Epidemic poliomyelitis began at the end of the 1800s. And by epidemics, of course, we mean larger numbers of cases, not individual cases here and there. The first one in Sweden in 1868, 14 cases, and then also in 1881. The first in the U.S. was in Vermont in 1894, 132 cases, and that was studied by the local coroner. And he made some very astute observations about the disease, which you can find in the literature, which have helped us understand the nature of it to this day. An important observation was made by a Swedish physician in 1887 who noted an initial systemic phase of the disease, that is generalized symptoms, fever, malaise, aches and pains, and involvement of the central nervous system, that is the paralysis, seemed to be a complication. He was very astute. He noticed it didn't happen in everyone, and so he said maybe it's just a complication, and we will get back to this, of course, a bit later. We now call polio a disease of modern sanitation. So I've just told you around the turn of the 20th century it began to emerge in epidemic form, and we believe this is because of the improved sanitation that was developed around that time. So before that year, polio circulated freely in the population. It's spread by the fecal-oral route, as we will see, and it infected most children shortly after birth, and so they became immunized. When we improved sanitation, that delayed infection until later in life, and one theory is that the children at birth are given antibodies from their mother that protect them against the disease. So if these children are infected early on, they're protected, but they make their own immune response and become immune to the infection. If you delay infection till later in life, when they no longer have maternal antibodies, these begin to decline at birth and are gone after a few months, then you will get infection and disease. So that's why we call it a disease of modern sanitation. And there's some epidemiological data that have been collected over the years that support this hypothesis. The virus was isolated by Karl Landsteiner, who's also well known for studying blood group substances. He was a Viennese physician. He isolated the virus in 1908. And what he did was he took the spinal cord of a boy who had died of polio, he ground it up, and he filtered it. Now, at this time, we already knew that there were agents called viruses, and they were defined by their ability to pass through a very small filter. These were porcelain filters that were manufactured at the time, and we now know that they had a pore size of about 0.2 microns. So he found that if he filtered the extract of spinal cord through this filter, the filter would retain bacteria and fungi, for example. The viruses will pass through. You then take the material that passes through with viruses in it. He inoculated into 2 old world monkeys and found that they developed the disease, poliomyelitis. It's a good thing he used old world monkeys. If he had done this with a new world monkey, it wouldn't have worked because they're not susceptible, and he would never have discovered the virus. He took these monkeys who developed paralysis, he sectioned their spinal cord, and he saw lesions just like the lesions that can be seen in human patients of polio. So this was indication that he'd passed the agent. He suggested that it was caused by a virus. He was also able to pass it from monkey to monkey. He took the monkeys who were paralyzed, ground up their spinal cords, filtered them, and passed it from monkey to monkey. And in fact, for many years, this is how virus stocks were maintained. There were no refrigerators or freezers at this time. So to keep a virus stock, you kept it in an animal. You infected a monkey, you incubated it for a week or 2. When the monkey became paralyzed, you would sacrifice it, make an extract, and pass it to the next monkey. And this went on for quite a few years, and in fact, it resulted in selection for some very odd viruses with unusual properties being passed this way from monkey to monkey. So let's take a little diversion now. Look at the virus that Karl Landsteiner discovered. We're going to go into a little biology of the virus. We classify viruses in a Linnaean fashion, and we put them in families. And the families are ending in AE. And so the family that includes polio is called the Picornaviridae. This was a name given to the virus many, many years after its discovery, and it refers to the fact that they're very small, pico, and their nucleic acid is RNA. And now in this family, just so that you have a reference, there are a number of genera which in turn contain different kinds of viruses, different species of viruses, if you will. So here we have the enterovirus genus, which contains the polioviruses. There are also a variety of other viruses within this enterovirus genus. It was originally named because the viruses pass through your intestinal tract when they infect you. But a number of years ago, with the advent of nucleic acid sequence analysis, all these viruses were reclassified. And now we include in the enterovirus genus the rhinoviruses, which are respiratory tract pathogens. This is very odd to me because rhinovirus has nothing to do with the enteric tract, so to put it in an enterovirus genus seems odd, but we are now at the victim of the bioinformaticians who tell us where our viruses must be classified. So that's polio there, and there are many other genera in the picornaviruses with a variety of very interesting human and animal pathogens, but that's all we'll say about that for now. These viruses are quite small. They're 30 nanometers in diameter, which is on the small side for viruses. The smallest viruses are about 20 nanometers. The biggest are over 750 nanometers. And this is a photograph of poliovirus taken in the electron microscope. You can see they are spherical particles, and they have some surface definition. That is because the surface is irregular. In 1985, the three-dimensional structure of this virus was determined by X-ray crystallography. So what that means is that we could determine the position of every atom in this capsid. This is the shell that comprises the virus, in three-dimensional space. So polioviruses are made up of 4 different proteins. They're called VP1 through 4, and these are repeated 60 times each to form this shell. This happens to be an icosahedron, which is the best kind of geometric figure to make a virus particle. Of all the solids that we know of, only the icosahedron is used to build viruses. And we think that's because it's the best way to make a stable particle by repeating subunits. So you take 4 different viral proteins and you repeat them 60 times, you build this icosahedral shell, and that is the virus particle which you see here. And within it, of course, is the nucleic acid. This is shown in green here. It's RNA for poliovirus. And the nucleic acid, of course, has all the information to make new virus particles. So this is really just a shell. It's just a carrier to bring the nucleic acid from one host or one cell to another. Now, when the three-dimensional structure of this was solved in 1985, these were the kinds of pictures that we could make. These are generated on a computer. So you have thousands of coordinates in X, Y, Z space for each atom that make up this capsid. And you can build the structure, and it looks something like this. Of course, we've taken the liberty of coloring it because these viruses don't have color at that size. But the coloring is actually done to pass some information on to you. We have colored this particular view of polio in a way such that the atoms farthest from the center of this icosahedron are white, and those closest are black. So you can see here, for example, is a very high point on the virus particle. We actually call this a plateau, and around it is a very deep part. So this particle has surface features. And that's what you're seeing in this electron micrograph right here. And in fact, for polio and related viruses, this dark part, this depression, which is repeated 60 times on the particle, is where the receptor binds. The receptor binds into this deep depression. So we can learn a lot from these structures. We can learn where antibodies bind that protect you against infection. We can learn how these initiate infection to get into cells. So this is one of the first viruses to have its structure solved in 1985, and many others have since followed. Now, inside that capsid, or protein shell, is the nucleic acid of the virus, and that's shown schematically here. Now, this nucleic acid is about 7,500 bases in length. The bases, of course, A, G, C, U, are repeated in a way to make up the genome. And until about 1980, we had no idea what the sequence of those bases were. This was actually my project as a postdoctoral fellow with David Baltimore, was to determine the sequence of that RNA. And I did it by making a DNA copy of it and taking that DNA and putting it in a plasmid, which is a vector that will propagate in bacteria, so I could get lots and lots of DNA. And then we applied a sequencing technique to learn the sequence. And I just want to pass on an anecdote. It took me 1 year to do this sequence. Today, I could hand this DNA to a company and a few hours later have the sequence of all 7,442 bases for less than a few $1,000s probably. So 1 year of my life I spent doing, which now would not be necessary for anyone to do. But it was well spent because I had good conversations with everyone in the lab. Now this RNA genome encodes all the information to make polio. And these viruses express their genetic information in different ways. This one makes a very long protein from this RNA and then cleaves it with proteinases that are encoded by the virus. So this is the long protein here, and you can see all these cleavage sites, and what you get in the end are the individual proteins that the virus needs to make more virus particles. So here are the capsid proteins, for example, and then there are a variety of proteins involved in making more nucleic acid, essentially. That's what all these proteins do. And here's a very important protein down here at the very end. This is an enzyme that copies the RNA, makes more of it. So one RNA will get into the cell, and what comes out are thousands and thousands of new particles. So this is the sequence that I created after 1 year of those 7,442 bases. I can never resist showing it's a year of my life, so there you have it. And this is actually a figure I made way back in the 80s. So you can see I've put the amino acid starting from base 742, which is where the protein initiates. There's an amino acid triplet. And that's the long polyprotein that is made and terminates here at the 3' end. Now, this is all the information you need to make polio. If you can make this nucleic acid and drop it into a cell, that cell will make more virus. So you have to keep that in mind when we think about eradication, because no virus will ever be really eradicated because you can always make it again from the sequence. These sequences live on computers. They're not going anywhere. You can get this anytime from NCBI. So even if we eradicated polio, someone could make it the next day by simply synthesizing this nucleic acid and putting it into a cell. And that's in fact what I did next in the Baltimore lab. I had all these plasmids with full-length DNA copies of the polio genome in them, and I just simply dropped them into cells in culture, human cells in culture. All right? And those cells made virus. So here we have polio virus. You infect cultured cells and they make virus particles. What I did was to take the viral RNA. We can extract it out of the particle. We made a DNA copy of it. That's shown down here. I put that DNA into cells. Just a blind experiment. I said, what would happen if we put this in cells? And new viruses emerged. So that's a process called transfection. And this was an amazing result because it's the first time it had been shown for any animal virus. But more importantly, having a DNA copy enables you to manipulate the virus. You can change it as you wish. You could add genes, you could subtract genes, you could mutate them. You can manipulate DNA. You can't do that with RNA very easily. So this gave us the ability to make different kinds of polioviruses. And now this technology is used for all viruses, and you can create various types of viruses at will that express different things. So this is why I'm at Columbia University. On the basis of this nice finding, I gave a job seminar, and I was offered a position back in 1982, and I've been there ever since. And we've been working on this, using this DNA to address a number of problems about polio. A few more things about the biology of this virus we need to know to understand the history I'm going to tell you. This is what happens when polio gets into a cell. So the virus is shown up here, the little icosahedral particle. It binds to a receptor on the cell surface. All viruses need to bind to receptors on cells. They don't just get in. All right? And that receptor determines what cells can be infected. We actually, in my lab at Columbia, one of my first students, Kathy Mendelson, actually identified it and cloned the gene encoding it, and that will be important in a moment. Once the virus binds the receptor, it ejects its nucleic acid into the cytoplasm, and this is a process that's different for many viruses, but for polio, the cytoplasm is where it wants to be because its nucleic acid is RNA, and the first step that will happen is that this RNA is translated into protein. Remember, you make that long protein, and then you chop it up to make all the individual proteins that you need. The virus also needs to make more of its nucleic acid, and so what it does is take that initial RNA and it makes copies of it. So it makes a copy once and then again to make the right polarity, and then these eventually assemble with new proteins to make new virus particles. And then at some point, the cell bursts open, the cell dies, and a new crop of viruses is ejected. And this, for polio, takes about 6 to 8 hours. It's a very quickly replicating virus. And that is consistent with it causing an acute infection in people, one that progresses very rapidly. Now, what does this do in people? So this virus causes poliomyelitis. This is a disease we acquire when we ingest the virus. So we show it here. Here are free virus particles. You ingest them, and they can replicate in the epithelial cells that line your alimentary tract. So this is an enteric virus. It can replicate in your upper alimentary tract, or it can make its way into the intestines and replicate there as well. So the virus can pass through your stomach, which of course is acidic. It can pass through the small intestine, which is alkaline in the very beginning, and it has no problem whatsoever. It is incredibly resistant. And so this is one of the reasons why this is so easily transmitted in nature, is that it's a physically very resilient virus particle. So the virus enters these cells in your mucosal surfaces in your alimentary tract, and it reproduces in them following the scheme that I just showed you. It enters those cells, makes more virus particles, then those cells are destroyed. As the viruses replicate in these cells, they are shed into your intestines and eventually pass into the feces. All right, and that is how viruses spread from one person to another by what we call fecal-oral contamination. So somehow the virus in the feces gets into the mouth of someone else, and children are very apt to do this when they're playing or not being very hygienic, and that's one of the main ways that the virus is transmitted. In addition, as the virus is multiplying in these cells, it also gets into your bloodstream. As it destroys these cells, it can go beneath the mucosal layer, and just beneath here there are lymph capillaries, which are very permeable. They can take up the virus. The virus travels from the lymph system into the blood and establishes what we call a viremia, that is, presence of virus in the blood. So shortly after infection, you have virus in your bloodstream. When viruses are in your blood, they can get anywhere, and that's not a good thing. Polio virus doesn't end up everywhere. It ends up in only a few places, and one of them is unfortunately very serious. It's the central nervous system. From the blood, the virus gets into the spinal cord and eventually the brain, where it does damage. Most viruses, in fact, can't get into the central nervous system. It's a rare event. And for that reason, the CNS isn't terribly well protected. Our immune system has not developed to the same extent there as it has elsewhere in our body, because we have very few pathogens that can get in. But polio is one of them that can get in. West Nile virus is another one. When these viruses do get in, they cause big problems. So polio enters the central nervous system. We think it might enter through muscles and be transported by nerves up to the spinal cord. But of course, in the spinal cord, there are a variety of cell types. The most important one are motor neurons, the ones that control your muscle activity. And those are the ones that poliovirus replicates in. It kills them, and as a consequence, you become paralyzed. And typically, you have lower limb paralysis with polio, not always, but typically, and that is a consequence of the destructions of neurons in the lower portions of your spinal cord. But there are other forms of polio as well in which other neurons are destroyed throughout the spinal cord. So humans are the only known reservoir of this virus, as far as we can tell. No other animal in the wild has it. We don't know where it actually came from. And even if it's been around since the ancient Egyptians, we don't know where it came from, an animal of some kind. There are no precursors to give us any clues. But this is important because it's one of the requirements for eradication of a virus infection that there's only a human reservoir. As I said, it's spread by fecal-oral transmission. So poor sanitation will spread this very readily. And as I said earlier, if you improve sanitation, then you delay infection to later years. these infections peak in temperate climates during warm months. In tropical climates, they tend to occur throughout the year, but in climates such as we have here in the U.S., in the summertime was the typical time to have outbreaks of polio, and of course that's when the kids are outside playing, and one of the reactions was to keep kids inside during the summer months to try and stem the disease. There's one very well-known complication. It's called post-polio syndrome. This happens about 30 to 40 years after the initial paralysis. So there is a certain fraction of individuals who get polio who die, but many of them have limb paralysis only and can survive. And with physical therapy, they can be rehabilitated to the point of gaining their limb function. And in many of these individuals, 30 to 40 years later, they begin to experience paralysis of the same limbs again. And that's post-polio syndrome. Happens in about 25 to 40% of those individuals who originally had polio. It is not an infectious process. As far as we can tell, it's not a matter of virus being in there all those years and somehow being reactivated. We're not sure what the cause is. One of the possibilities is that the initial incidence of polio, the bout of polio destroyed a number of motor neurons in your body, and as you age, you lose more, and you don't have sufficient numbers to innervate the muscle. But we still have in this country a number of individuals with post-polio because we had many, many cases of polio in the 40s and even in the 50s. It's only until those individuals have died will this cease to be an issue. Now, this is a chronology of polio infection that I want to go over with you to give you a flavor for what happens with this disease. This is from a 1950s textbook of medicine. It's a hand-drawn figure, which I find very charming. You don't find these anymore in science. And it takes the course of a polio infection. We have days here on the top, the phase of infection and what's going on in terms of temperature, virus in various places, and also virus in the CNS. So let's track this through, and this will help us understand the disease. So here we have exposure to the virus starting here. For the first 7 days or so, you're well. There's no sign that anything is going on. And then after about a week or so, and that can vary, of course, you have a spike in temperature here. You have headache, sore throat, nausea. These are systemic symptoms. They could be almost any virus infection. They could be flu, in fact, so you can't really tell that this is polio. At the same time, you have virus in your throat. You can see the rise of virus in throat secretions. It's this curve, and here's virus. Throat secretions here, virus in the feces. So very early on, when you have these initial symptoms, you have virus in your throat and feces. So you're already shedding virus. But no one knows that you have polio. This is very important because this is how it spreads so effectively. There's no way to know that a person coughing with a sore throat has a polio infection until much later, and by then the virus has spread to others. At this time, when you have this initial systemic bout, you have virus in the blood. You see there's a peak here. It peaks and then goes down. Okay, so in most of the cases, 99 out of 100 cases, that is the end of the infection. You will recover, and you will never know you had polio unless you look for antibodies to the virus, which you can find later in the serum. But in about 1 out of 100 or maybe 200 infections, the virus somehow gets its way into the central nervous system. Remember, it goes from your intestine to the blood into the CNS, and this bottom graph is virus in the CNS. And then at some point you start to develop stiffness, pain and paralysis, and that is the typical poliomyelitis. You can have paralysis of the leg muscles, as I've said. You can also have respiratory muscles, which is a more severe problem, and also paralysis of nerves that innervate your heart as well, and that can be fatal. So it's a very rare invasion of the CNS, and that's why that original individual, Meaden, in the 1800s, said he thought that the paralysis was a complication because it wasn't very common. Now, in our lab, and when I started my lab at Columbia in 1982, I wanted to study this disease. I had spent 3 years sequencing polio and studying it in cell culture, but I was really interested in this disease. As you can imagine, I had read about it. I had actually met Albert Sabin, who developed one of the vaccines. I wanted to understand how this disease developed, but there was no way to do this because the virus only infects people or chimpanzees. And I couldn't afford chimpanzees, and I didn't really want to work on chimpanzees anyway. So I said, could we get a mouse to be a model for polio? So as I told you earlier, one of my first students had identified the cell receptor for polio. So this is a protein on the surface of cells to which polio attaches and that lets the virus get in. And we had found that if we took this receptor and put it on mouse cells that we were growing in a culture dish. Now, these mouse cells, if you put polio on them, nothing happens. The virus doesn't get in. But we found if we put this human receptor into those mouse cells, the virus would then get in and replicate and kill the mouse cells. So I was very young and naive, and I said, let's do this in a mouse. Because afterwards, years later, people told me that was a really bold experiment, but at the time, I just thought it would be interesting to try. So we made what was called a transgenic mouse. We took the gene encoding this receptor, we put it into the chromosomes of mice so that all the mice born subsequently would have this gene, and then we infect them with polio, and you can see they develop paralysis. This mouse here has a paralyzed left hind limb. Beautiful recapitulation of the disease. In fact, here's a section of the spinal cord from one of these paralyzed mice. This is a lumbar section, so it's lower down in the spinal cord, innervating the lower limbs. And what we have done here, well, first of all, before I show you where the virus is, see, all this is stained in a certain way to emphasize the presence of specific cells. You can see there are large clumps of cells here and here and here. These are actually immune cells coming into the spinal cord to try and get rid of the infection. So this spinal cord is infected. And we can tell that it's infected not only by the presence of these immune cells, which you would never see in an uninfected mouse, but also from these green dots here. And what we have done here is we have stained this section with a probe that will detect viral RNA. So all these green dots are poliovirus RNA multiplying in these cells. And these happen to be neurons. We can tell because they're very large and they are pyramidal in shape. And that is where polioviruses replicate. There are many other cell types in the spinal cord, but polio doesn't replicate in them. Only the neurons and only the motor neurons, and that's why you get paralysis. So in this mouse model, you can inject the virus, say, into the tail vein or intraperitoneally. It will make its way to the spinal cord. It will destroy neurons, and that's why the mouse's hind limb is paralyzed. So we could use this model to study all sorts of questions about polio, how it caused disease, how it traveled in the mouse. And we also used it to study Albert Sabin's polio vaccines, which we'll get to in a moment. And these were the vaccines that were used to eradicate polio in the US and are now being used in the eradication campaign in the rest of the world. So it was a very fruitful model for us to develop. And I have to tell you, just 2 days ago, I destroyed the remainder of this colony in my lab. No one else is interested in my lab and working on them any longer. It's an expense to keep mice, so they're now gone. But we worked on them for 22 years, and they're no longer around. Okay, let's get back to polio history and now move to larger and larger epidemics. The first big one in New York City in 1916. Not just in New York, but in the Northeast. 9,000 paralytic cases in New York City, 27,000 total in the Northeast. And remember, you have to multiply this times 100 to find the total people infected, because only 1 in 100 or 200 people are actually paralytic. You have a lot of people walking around who are shedding virus. They have some cold-like disease, but they're not paralyzed. And that's why again, why the infection spreads so readily. In this epidemic, this practice of quarantining was used. This was something that was developed in the preceding years, and they said, well, if you have someone who's paralyzed, we're going to put them in quarantine so they can't infect anyone else. And at this time, that was realized to be futile, because during this epidemic, we learned that, in fact, most people who are infected with the virus don't show paralysis. So there's no point in quarantining the paralyzed individuals, because for every one, there are 99 walking around who are actually shedding the virus. This is a problem with many virus infections, not all. Smallpox, for example, every infected case shows symptoms, and that's one of the reasons why you could eradicate it the way they did, which was to find a case and then do ring immunization in that area. But you can't do that with polio because you can't see everyone who is infected. And that will return later as an issue in polio eradication. So from this epidemic, we had 4 conclusions. First of all, it's exclusively a human infection. There was no evidence for any other animal being involved. It's far more prevalent than apparent from clinically recognized cases. Okay, 1 in 100 or 200 paralytic cases per total infected. And these asymptomatic in mild cases, so asymptomatic, no symptoms, or the mild ones, that is the systemic sore throat, cough, etc., those are the most important in spreading the disease. As a consequence, if you have an epidemic where you have 1 to 3 cases per 1,000 people, this will immunize the general population. That's why epidemics are self-limiting, because you have enough infection without disease that the virus is spreading to everyone, and it basically immunizes everyone. So every polio epidemic is self-limiting, and it will only occur again when you have enough susceptibles in the population. So these were very important lessons learned. Now, many of the photos of polio from those eras show people in iron lungs. There are many, many books on the history of polio. This was a very popular topic to write about. It was a very scary and prevalent disease in the U.S. These you don't find anymore, but when individuals had polio paralysis of the respiratory muscle, they were put into these iron lungs, which did the breathing for them, and most of them could come out. There are a few individuals who were in these for their entire lives, though. And, there was an incident a few years ago, a woman who had been in since the 1940s, and she died in the iron lung. But hospitals now no longer have these, of course, but very common photos. Now, one of our presidents, Franklin Roosevelt, of course, is believed to have polio, which is another remarkable thing, I believe, in that a U.S. president being elected with the paralytic disease, of course, wouldn't happen today. But back then, there was no internet, not much television, and so it was possible. He, of course, wanted to find a solution to the disease, so he started the March of Dimes, which raised money, and eventually that money was used to develop polio vaccines. And along the way, it took many years to make a polio vaccine. This is a good lesson, actually. So the virus was isolated in 1908, and the first vaccine that worked was released in 1954. It took 50 years to make a vaccine, because we had to learn a lot of things before we could make a good vaccine. Not that people didn't try. People did try to make vaccines, but they didn't work. So here are some of the milestones in leading to a vaccine. 1931, the isolation of polio from throat washings of people who were not paralyzed, right, the throat. And people, again, realizing that this could be spread from people who didn't have paralytic disease. 1941, isolation of virus from the pharynx, the ileum, and the nervous system of people with polio, but not from the nose, the olfactory bulbs. For many years, people thought that this virus was spread from the nose to the nose of someone else. The virus entered the nose and entered your brain through the nose, and that was incorrect. 1947, virus was isolated from sewage, which told us that it was probably passed by fecal-oral routes. And finally, in 1954, the finding of virus in the blood. Again, all this is a contributor to the pathogenesis, which I have told you. A big discovery in 1949 by John Enders and colleagues, being able to grow the virus in cultures of human cells from non-nervous tissues. This was the first growth of a virus of any kind in a cell culture, and it replaced the monkey from detecting and propagating polio. And this allowed us to grow a vaccine. You can't grow vaccines in animals, so we needed a cell culture, and this was the key discovery. And they got the Nobel Prize for that in 1954. A study of Arctic Eskimos showed that a single subclinical infection, that is without paralysis, gave you immunity which would last for at least 40 years. So that said, we can make a vaccine. If we can make antibodies, then we can protect people from polio. And finally, in 1951, just a few years before the vaccine was released, we learned that there are 3 serotypes of polio. Now, a serotype means that if you make an antibody against one serotype, those antibodies will protect you against that, but not of any other serotypes that may exist. So people didn't know this earlier, that there were 3 serotypes of polio, and they would make a vaccine against one serotype, and then the other 2 serotypes would come in and cause polio. So this was a very important finding. The first vaccine to be released was inactivated polio vaccine, or IPV. This virus was grown in cells using John Ender's methods, and its infectivity destroyed by treating it with formaldehyde or formalin. So this destroys the virus particles so it cannot be infectious. And that was developed by Jonas Salk. It was subjected to a clinical trial in 1954. The National Foundation for Infantile Paralysis, the March of Dimes, founded by FDR, funded this clinical trial. 1,800,000 children, the biggest ever medical clinical trial in kids. The vaccine was 50% protective. It was released. These data were released on April 12, 1955, and the vaccine was licensed on the same day. So this was met with great fanfare in the U.S. And here are some of the headlines from New York newspapers at the time, which show that this disease was a big deal. And these are all the papers that used to exist here in New York, I guess, with the exception of the Post and the Mirror, are the Times are the only one left. But I remember all of these when I was a kid. However, this is an interesting coda here that shortly after this vaccine was released, within 10 days, kids who got it started to develop paralysis. All right? And that was eventually linked back to the vaccine itself. Eventually, it was determined that 200,000 people were infected by this vaccine. 70,000 became ill. There were 260 vaccine-associated cases in 94 immunized kids and 166 contacts of them, and a number of deaths as well. The reason why it happened is that one of the companies who had made the vaccine, there were a number in the U.S. who were licensed by the FDA to produce the vaccine. One of the companies, called Cutter Laboratories in California, hadn't properly inactivated the virus with formaldehyde. So they actually sold the vaccine with infectious virus in it. And this is injected into the kid's arm, and they got paralyzed. Now, can you imagine, here you're a parent waiting for this vaccine, and you bring your kid to get immunized, and then 10 days later they're paralyzed. So this is very sad. This, according to Paul Offit, is the beginning of the anti-vaccine movement in the U.S. because a huge lawsuit resulted as a consequence of this, and the lawsuit established product liability, that no matter what a manufacturer does, if someone gets hurt, someone has to be liable for that. It's a very interesting case. In fact, Paul Offit wrote a very good book about this called The Cutter Incident. Nevertheless, it was restored and corrected and returned to use. And in the U.S., polio fell from 20,000 cases a year to 2,500 by 1960. But many people felt that this vaccine was not the one that was going to eliminate polio from the U.S. They felt that we needed a vaccine that would mimic a natural infection. And there was some data to support this. Hilary Kaprowski and John Enders in the early 50s showed that if you took polio and just passed it in cell culture, from cell culture to cell culture, you could make viruses that had reduced virulence in animals. So the model used to study polio in these years was the monkey, various types of old world monkeys. And they could show just by growing the virus in cell culture that the virus would be less lethal for animals. So Albert Sabin took advantage of these observations, and he developed what we now call the oral polio vaccine, or OPV, Sabin's vaccine. And of course, he made all three serotypes, as did Jonas Salk, because that's what you need to protect. These vaccines were licensed in 1961. Sabin had a hard time getting them licensed in the U.S. because we had the Salk vaccine already, and the U.S. Public Health Service said we don't need another vaccine. So Sabin went to the Soviet Union, which existed at the time. He had a friend there. He had come from Soviet Union originally. He was born there. And he had a friend called Chumakov, who was the father of Soviet virology. And he convinced him to do a clinical trial with his strains. They put it in 100 million kids, and Sabin came back with the results and got his vaccine license. Very interesting story. He carried out what were called Sabin Sundays, community-wide administration of his vaccine, even though still, until the 80s, only 60 to 70% of kids were getting the 3 doses. So in the beginning, you would give type 1 first, then type 2, and then type 3. So you had to have the kids come back 3 times, which is very difficult. You're never going to get 100% vaccination when you have to keep coming back. So not everyone was getting immunized. Eventually, it was incorporated into routine vaccination program for young children, so now you have to be immunized in order to go to school. And that brought cases from 2,500 when only IPV was used to 0 in 1979. That was the last indigenous case of polio in the US. So those are the two vaccines that were made. We still have both of them. You can use either one depending on the country that you're in. We have inactivated vaccine. This is an injected vaccine, by the way, so you need a trained health care person to do it. If you make it right, it doesn't give you polio. And since the 1950 incident, it's been fine. It doesn't give you immunity in your intestine. It's injected into your arm, gives you antibodies in your blood, but you could still ingest polio, and that would replicate in your intestine. It would get into your blood, and then it would hit those antibodies and be neutralized. But you could still excrete polio. Okay? The reason I say this is because the US now is using only IPV. So the collective alimentary tracts of Americans are susceptible to polio. So if a strain came into the US, it could replicate in our guts and perhaps spread to someone who wasn't immunized. So we used IPV until 1961, and then we started using it again in 2000, and we'll talk about why. OPV is very easy to administer. You just drink it. You don't need any doctor or nurse to do that. You get intestinal immunity because it replicates in your gut. And these are made of viruses that Albert Sabin empirically derived from wild-type virus. We'll talk about that. And a very important point, it usually reverts in the intestine as it replicates, and we will talk a lot about that. And we used this from 1961 to 2000. In 2000, we switched to IPV. So how do these vaccines work? Again, you ingest polio, it goes into your alimentary tract where it replicates. OPV will block the virus from replicating in the epithelium of your alimentary tract. IPV will not. It will only block virus once it enters the blood. So two very different vaccines. And this is the effect on polio in the US. Here, the number of cases annually, 20 to 30,000 in the US. An introduction of the inactivated vaccine caused a precipitous decline in the number of cases. And then we brought in the Sabin vaccine in 1961, last case in 1979. We have no more, for the most part, no polio in the US with a few exceptions. Nevertheless, even though we had two really good polio vaccines, in developing countries, most of the kids didn't get them for a variety of reasons. And so there was still a lot of global polio, even by the 1980s when we had eradicated it from the US. Now, Sabin went to Brazil at this time, and he instituted mass immunization campaigns, which he called National Immunization Days. And he showed that you could eradicate polio in a country like Brazil by just 2 immunizations. And as a result of his work in Brazil, the WHO established this expanded program on immunization, and they said, we're going to help increase the use and distribution of polio vaccine worldwide. And it's really because of Sabin and WHO taking this initiative that we're at the point where we are today. So here's the data from Brazil. These are the incidents of polio cases in Brazil before immunization. These are the number of cases here. And you can see he did twice a year, June and August every year, and eventually got rid of polio in Brazil. Very important. So the WHO eventually decided that they could eradicate polio, and I think you've heard about this in this series before. They made a resolution in 1988, and the idea would be eradication would occur by 2005, and then at some point we would stop immunizing, because you don't want to keep immunizing for a disease that doesn't exist. So here we are in 2012, and we still have polio. So let's look at why and how that relates to the biology of the virus. So here's the incidence globally of polio, 1988, 600,000 reported cases globally, probably vastly underreported, much lower in 1988, 1998, and in 2008. And I'll show you the statistics today, less than 1,000 cases per year. So here are the challenges to eradicating this virus now. We still have transmission of wild viruses that are out there in certain places. We have a complication that we didn't predict. They're called vaccine-derived polioviruses. We'll talk about that. We have persistence of vaccine virus in people without antibodies. And then we have reinfection of previously polio-free areas. So we're going to touch on some of those in the next few minutes. So how did Sabin make his vaccines? It's important for us to understand this. And this is an example of the type 3 component. Remember, there are 3 serotypes. Type 3 was isolated from a fatal case in Los Angeles in 1937. Sabin took this. He passed it extensively in monkeys. So passage means you infect the monkey gets paralyzed, you grind up the spinal cord, you make an extract, you pass it to the next monkey. He did this 21 times here, then he passed it in monkey testicle cultures, monkey kidney cultures, 39, 3. So he just kept passing it until he got viruses that would no longer kill monkeys when he put them right in the spinal cord. So it's an empirical process. And he eventually got vaccine strains that actually worked. And here's one of them. He did this for all 3 serotypes. And then when he had them all, this was 1950, what do you think the first thing he did with them was? Do you think he got IRB approval to do something? You know what IRB is, right? Institutional Review Board. What do you think he did? He ate them, exactly. He drank them, actually gave them to his wife and daughter, as well as himself. They're all seropositive because they had gotten antibodies by inapparent infections, but he tested them on himself. That's the way it was done those days. Then he gave it, who did he give it to next? Prisoners, volunteer prisoners. And then he moved into larger clinical trials, and eventually the Soviet Union, 100 million kids. And that's how it got licensed. But today, of course, you don't do that. So when I started working on polio, we had the ability to sequence the genome. First time in the 1980s. So we could say, let's take what Albert Sabin made and sequence it and compare it to what he started with and see what are the changes. So we did that, and there were many, many changes from the virulent to the attenuated virus, that is the vaccine virus. But then we took my mouse model for polio, and we put the changes in one by one into the virulent virus and said, which one of these make it a good vaccine? We did that for many years, and other people did it in monkeys. And this is the result of that for all 3 serotypes. These are the mutations in the genome that Albert Sabin selected for, which are very important for making these good vaccine strains. So these don't mean a lot to you. You can see the type 1 has 5 changes, and then the type 2 and 3 have 2 changes that make it a good vaccine, just 2. So we didn't know this when Sabin's vaccines were licensed, because if we knew it, they would never have been licensed. Today, if you tried to license a vaccine, a live attenuated vaccine with 2 changes out of 7,500, the FDA would say, come back when you have, I don't know, 100 or 50, or it's more stable. But we didn't know that, and technology gave us the ability to know this. So very few changes make Albert Sabin's vaccine work well. And I want to show you where some of these are. You see, all 3 viruses have a change in what I've labeled UTR. This means untranslated region. This is a part of the RNA that is not translated into protein. And it's way at the very end of the RNA. So remember the schematic of the RNA of the virus. There's all this green part in the middle codes for protein. The very end here, we call this the 5' end. It has an untranslated region. It's orange. It's not translated into protein. That's where those mutations that Albert Sabin selected for are located, in this very interesting region that has a lot of RNA structure. And you can see they're all in the same area. This is the change in the type 1 virus, the type 2, and the type 3. And this is amazing to me because he selected these viruses separately in monkeys, but they all end up having a similar mutation in a very, very neighboring part of the genome. All right, so this is probably the most important mutation in all these strains that make his virus not virulent when you drink it. That's important to remember. So in my lab, we had made these mice susceptible to polio. So what we did is we infected them with 2 different polio viruses. And one of them has a base at 472 that's in that 5' UTR. It's a U. That's the base that was in Sabin's polio vaccine. The other virus has a C at that position. That is the base in the virus that Sabin started with. That's the virulent base, if you will. If you inject, so these 2 viruses are identical for 7,440 nucleotides, except for this one base difference. If you put them in mice, this one doesn't do anything to the mice. You can put as much virus as possible that you can inject. None of them get paralyzed. They're fine. If you put in the C, if you put 9,000 infectious viruses into them, half the mice get paralyzed. So this is quite lethal. And just changing one base makes a big difference. So this is an incredibly important base, and this is why when we drink the Sabin vaccine, we're not paralyzed. If you look at the growth of this virus with time in the brain of these animals, so we're measuring virus here and we're measuring it with time, you see the viruses with the U don't really grow. They eventually are cleared from the CNS. So those are the ones that don't cause paralysis. In contrast, the virus with a C grows very well in mice, and that's why it paralyzes them. So this just emphasizes the importance of this base change. And this is going to come back to haunt us in a moment. Now, in the 1950s, when Sabin was first testing his vaccines in his first volunteers, what he would do, he would feed some volunteers his candidate vaccines, and then he would collect what came out in the feces. And then he would study that. And what he found was that, and others found, that some people who received these OPVs, they excreted viruses that were more neurovirulent than what he put in. Neurovirulent meaning causing paralysis in monkeys. So that was the first clue that something was going on with these viruses as they replicated in the individuals. When his vaccines were licensed in the 60s, there were cases of polio associated with type 3 vaccine in Eastern Europe in the early 60s. And we now know these were caused by the vaccine itself. So very early on, we knew that Sabin's vaccine would cause polio. And as we eliminated indigenous polio from the US, that is polio caused by circulating virus, the only paralysis left was caused by the vaccine. We call this vaccine-associated paralytic polio, or VAP, and it occurred at a rate of about 7 to 8 every year in the US. And it's about a rate of 1 per 1.4 million doses of vaccine. So for every 1.4 million kids who got the polio vaccine, one of them would be paralyzed. Or sometimes the parent would be paralyzed by the virus excreted from the child. Now, as a result, we switched to Salk's vaccine in 2000. So there was no more polio in the US after 1979, except polio caused by the vaccine. So you can imagine this was an intelligent public health decision. By this time, of course, Jonas Salk had died, so he didn't get to see his vaccine resuscitated again. But we switched to IPV for that reason, because OPV, Sabin's virus, which had eliminated wild polio in the US, was causing a paralytic disease. So here is a detailed view of the cases of polio in the US from 1961 to 2003. Here's an overall view. You see going from 10,000 in the mid-50s, the introduction of IPV, and then in 1961, OPV, and eventually eradication. Now let's blow up this shaded portion, and we'll look here, starting in 1961. Total cases is shown by the line, and vaccine-associated cases by the bars, the dark bars. So you can see here there were a number of vaccine-associated cases, but they are overshadowed by the wild polio. But as you eliminate wild polio, you see the only polio in the US is now caused by the vaccine, 1973, 5, 76. Every case from then on is caused by the vaccine, except for 1979. This was an outbreak in an unvaccinated Amish community in Pennsylvania. This was virus that had been imported from Turkey. So after that point, there is no more wild polio in the US. So in 2000, we switched to IPV, now 0 polio in the US. Although there was recently a case which we'll talk about later. Now, why is this happening? Why does polio vaccine cause polio? I've given you some clues, but let's look at some more data. In 1985, Phil Miner, a friend of mine from the UK, he worked for the FDA equivalent in the UK. He was responsible for testing and release of polio vaccine. He had a baby, and the baby received polio vaccine. And he was aware of the work I've just told you, where we identified the mutations in the vaccine strains that made them good vaccines. And so he said, is it possible that these are changing in the gut, and can we pick it up? So after his child was immunized, he had his wife give him every diaper that the child produced for the first month or so of life. And he brought it into the lab, and he scraped off the feces and isolated polio and sequenced it. It's called the nappy experiment. It's famous in virology. And he published a Nature paper based on this in 1985. So here are the data. His son is DM, David Miner. And on day 24 after immunization, the virus that David was excreting, it's polio vaccine virus, it has a U at 472. That's the base that's very important for making it a safe vaccine. That's the one that I studied in mice, and I showed you before. Now, the vaccine, of course, also has a U, so that's good. DM2 at 31 hours, still a U, very good. But look, at 35 hours, it's now a virus with a mixture of U and C at 472. And by 47 hours, 2 days after receiving the polio vaccine orally, David Miner is excreting virus with a C, which, as you know, in mice will paralyze the mice. And that he does for the rest of, he excreted this for about a month. And you can see that these viruses are virulent. So the way we measure polio virulence, we can, of course, look at paralysis in an animal, or you can take the spinal cord and look for lesions caused by polio, that is, killing of cells by polio replication. And that's where this histological lesion score is. It's very low for the vaccine, but you can see as with increasing time, David was excreting virus of increased virulence. So these two lesion scores would cause this to fail the FDA tests for being released as a vaccine. Okay, so he's excreting very virulent virus, but David Miner was fine. And in fact, this happens in every kid that gets polio vaccine. After 2 or 3 days, they excrete reverted virus that is fully virulent. Most of them are fine, but 1 in 1.4 million are not. They get paralytic disease. And this is why, because in your gut, you select for viruses with a C. The C viruses, which contaminate the vaccine prep, you can't avoid it. They are selected for, they grow better, and within just 2 days, they dominate the population. So that is a problem if you are using this vaccine to eradicate polio. So polio vaccine, I think you will recognize, is very different from smallpox because the polio vaccine can revert to cause the disease it's meant to prevent. Smallpox doesn't do that. Smallpox vaccine is very different from smallpox virus. It doesn't revert to cause smallpox. So when we use OPV to eradicate, which is what we're doing now, the only source of polio is the vaccine once you get rid of wild virus like we did in the US after 1979. And that's why WHO in 1988 said we're going to stop immunizing at some point when we eradicate polio because we don't want to have vaccine-associated polio. The assumption there was that all of those vaccine-derived strains which you generated by immunizing millions and millions of kids who then excreted those reverted viruses into the sewers, the assumption is that they will not last very long. They will go away, so you could stop immunizing and they would not pose a threat to any new babies who were born and weren't immunized. Okay, that was the assumption WHO made, and they turned out to be very wrong about that. And the reason was there were, in the 90s, a number of outbreaks in a variety of countries, starting in the Dominican Republic, Haiti, but also in Egypt, Philippines, Madagascar, all outbreaks caused by vaccine-derived polioviruses. These were in areas where immunization coverage dropped, so there wasn't much polio. There was failure to immunize sufficient numbers of people, and the virus somehow got into these populations and caused outbreaks. But it was vaccine-derived virus. We can tell that very easily. We sequence it. We can see that it's Sabin-derived. So these are vaccine-derived viruses that regain virulence, as I've told you what happened to Phil Miner, his virus that he excreted, and they also can spread in human populations. In addition, we have many people in the world who have immunodeficiencies, in particular people who lack B cells, they can't make antibodies. You can't tell that they lack antibodies at birth. So shortly after birth, you give them polio vaccine, they can excrete it for years. It's been documented that these individuals will excrete polio up to 15 years. It's another reservoir of infection. And in fact, many of these strains have come from immunocompromised people to cause these outbreaks in these areas. And so this is a summary of some of these outbreaks. First one is in Dominican Republic, Haiti, 21 cases in July of 2000. So you can isolate virus from these individuals, you can sequence it, and then you can say how many years it's been circulating in humans. because polio changes. When polio is circulating in the population, it changes at a rate of 1% a year. It has a very steady clock. So you can count the number of changes in any isolate that you get, compare it to the Sabin strain, and say how many years this virus has been circulating. So here you can see this, the virus that caused this outbreak in Hispaniola circulated for 2 years before it caused the outbreak. And it's thought that this came from an immunocompromised person who was harboring the virus for many, many years. Now, there are 21 cases of paralytic disease, but remember, the number of infections is much greater. And in this case, it was estimated to be 1 to 200,000 total people infection. So 21 cases may not seem like much, but you always have to remember that there are many, many more people infected, and they can spread the virus to other people as well. So even today, if we have 1 case of polio in India so far this year, which is the case, there could be 100 or 1,000 people infected with the virus still. And these are all other outbreaks that have occurred. Similar idea, virus circulating for many years, causing an outbreak in under-immunized populations, all vaccine-derived isolates. Right? So this trashes the WHO idea that we can stop immunizing, because they assume that all these vaccine-derived viruses would go away the day you stop immunizing. But they don't. They circulate for years. So you can't just stop immunizing. And this is an interesting case in the US, which I wrote about not too long ago. This is a case of polio just recently. And this is a woman who died of polio in Minnesota, 44 years old. She went to the hospital with symptoms of muscle pain and paralysis. She was in the hospital for 3 months, eventually died. During her stay in the hospital, they had no clue what was wrong with her, but eventually they isolated polio from her and sequenced it and calculated that she had been harboring it for 12 years. So this woman has no antibodies. She's B-cell deficient, and she probably got this virus when her kid got polio vaccine 12 years ago. And she got the virus, replicated in her for 12 years, and then for some reason recently it went into her CNS and caused paralysis. Now these individuals get weekly injections of antibodies to protect them against infection, but this didn't help her. So apparently she got colonized. So this is an example of what can happen with these immunodeficient individuals. They can harbor virus for a long period, and if there are people around her that are not immunized, they can be infected as well. So what this means, to summarize, that these outbreaks show that these strains of OPV can circulate even in immune populations, and in many of those outbreaks they were circulating in immune populations. And as I said, if you stop, this mimics what would happen if you stop immunizing. You're going to have circulation of these revertants, which will infect new children who aren't immunized. So I don't think you can stop immunizing as WHO originally proposed. So after we eradicate polio, you have to immunize against OPV, essentially. I think you have to switch to the Salk vaccine globally until all the circulating vaccine-derived viruses go away. Now, this is a problem because IPV costs more than OPV. OPV you can make for about 5 cents a dose, and IPV is about $1 a dose. But we can just get more money from Bill Gates et al. The real problem is that these inactivated vaccines have not really been shown to be effective in certain tropical underdeveloped countries. So it's a bit of an experiment. We may have to develop a vaccine that is, in fact, better. But I think this makes the most sense to switch to IPV, and then you have to monitor the environment to be able to know when there's no more circulating vaccine-derived polio present. And so there's a good example of this was done in New Zealand. They switched from OPV to IPV in 2002, and they monitor the presence of polio in 3 different sewage treatment plants in the country. So here we have % positives for poliovirus with time, starting in 2002 through 2003. So you can see that the amount of OPV, this is a circulating vaccine-derived virus, within a year basically declines. There's still a little background. This is an imported OPV, so they could take this virus and sequence it and say it's OPV. And we haven't used OPV since the previous year, so it was probably imported. And eventually you detect no virus. So this is what I would propose. Globally, you switch to IPV and then monitor the environment for the presence of vaccine-derived viruses. And at some point, you have to make a decision we stop immunizing. I don't know who will make that decision, but it's a pretty important one to do. So a number of years ago, I forget when this was, early 90s perhaps, I was at a meeting in the National Academy, and I spoke about this whole issue, and I said we should switch to IPV. And D.A. Henderson was there, and I think he talked to you in this series, and he got up and he said, so we have then, say, 7 or 8 years, whatever it is, to gear up to the vaccine, then another 10 years to use inactivated. This is just totally unrealistic. We are not going to have the financial support globally for this. There's no way it's going to come about. And as an endgame strategy, it is dreaming to believe that this is reasonable. So it is just not on. So I was surprised because D.A. Henderson eradicated smallpox, so he must know, right? But now the WHO has decided that we need to transition to IPV, so it's going to happen somehow. So the moral is that when someone speaks forcefully, always look for what is behind that, I think. So D.A. Henderson now says, yeah, we have to use IPV, but when I got up and suggested it, he said it's not going to happen. So I thought that would be interesting to hear since he did talk to you guys. Okay. So what other problems do you have? You have vaccine-derived polioviruses. You have reintroduction, or you have reintroduction coupled with governments not wanting to immunize. 2003, rumors of contaminated OPV, which is made in the West, led the government of Nigeria to suspend immunization. They said, we don't trust this. So for 1 year, they didn't immunize any kid. So what happened? There was still wild polio in the world. There was an outbreak. Spread, and not only did the virus, I think it came from Indonesia and India to Nigeria, had many, many cases of polio in Nigeria, and then spread to neighboring countries in Africa where they had already eliminated polio as a consequence. So three outside Africa as well. So they've decided a year later to start immunization, but we still have endemic polio in Nigeria, 388 cases in 2009. And so far this year, just 17, but remember, multiply it by 100 at least for the number of infections. Now, to continue this interesting story, this is another example of the complications we have. In 1999, WHO said there's no more type 2 polio globally, it's eradicated. So when Nigeria started to resume immunization in 2004, they used a vaccine with just type 1 and type 3 components. So what do you think happened? I'm sorry, I didn't hear it. What? What happened? The type 2 outbreak caused by what? Where did the type 2 virus come from? Not where, but what kind of virus? Vaccine-derived type 2, right? So you should not have switched over. It's another example of what happens. If you stop immunizing, there's circulating vaccine-derived polio. You can't stop unless you make sure they are not there. So vaccine-derived type 2 caused this outbreak in Nigeria. So you really can't consider type 2 eradicated if there's still vaccine-derived type 2, and it's still used in some parts of the world, the type 2 OPV. So here's the situation at the moment. I will wrap it up very quickly. 2011, I just got these data yesterday. You can go to polioeradication.org and you get daily numbers on the eradication effort. So these are all the countries in yellow where we still have polio. The ones with solid yellow we call the endemic countries. We have never gotten rid of polio from these countries: Nigeria, India, Afghanistan, and Pakistan. And then the others, which have little hatch marks, these are importation countries. So these may or may not have been free of polio at one time, but then it comes back in. And the reason it comes back in is because it's still present in these other countries, and global travel brings it. And do you think there's a paralyzed person sitting on an airplane bringing the virus? No, it's one of the people who's just shedding virus but has a cough or maybe no symptoms. So that's why it's so easy to spread. So here are the numbers. Last year, 2010, globally 1,349 cases of polio, 232 in endemic countries, over 1,000 in non-endemic countries. These are introduced cases from other places. So you multiply this times 100 to get the total number of people infected. So 1,000 looks like a good number, but really 134,000 people are still shedding virus. The point is you have to be vigilant. Even if you get the cases down low, you have to keep going in and immunizing, massive campaigns, and that may not be so easy to do. This year we have, to date, 252 cases, which is pretty good, 84 in endemic, 168 in non-endemic. So we're doing a little better than the same time last year. So this is good. It looks pretty good. And here are the numbers for all the countries where cases of polio were reported. And I just want to point out a couple of things. Here's year-to-date 2011. So India, which has been one of the toughest countries to bring down the numbers, it's been at 1,000 or so cases for the last 5 years. Suddenly now we have one case so far in 2011, 2010, 22. So that's really good. But the monsoon season is now coming on, and during that time there's a lot of transmission, so we'll see what happens. Again, remember, there are at least 100 people infected, so you have to keep going in and immunizing. That's still a lot of cases in Pakistan. Pakistan and Afghanistan have a problem because it's sometimes very difficult to get in and immunize. There's armed conflict going on, and the teams that have the vaccine often cannot immunize kids. And so they don't get vaccine, then you have outbreaks of polio. There have been some outbreaks in Africa this year. You can see DR Congo and Chad had a number of cases. These were all imported from other places. And also last year in Tajikistan, there were quite a few cases of polio imported from India. So this emphasizes that you can get the numbers down very low, but you have to keep hammering with the immunization. You can't leave pockets open. But it's a problem, of course, if there is a war. And in places like the DR Congo, there's a lot of unrest, and it's very difficult to immunize everyone. So we have a great vaccine that'll definitely eradicate, but it's often hard to do it. And remember, even if we eradicate polio from the globe, as long as we have the sequence, you can make it again. So I think it may be possible to eradicate polio, but we do have that sequence that I made years ago sitting in computers, and if someone wanted to, they could make it and reintroduce it. So we have to stockpile vaccines after eradication and be on the alert for something happening. And I want to leave you with just one more thought. What vaccine should we stockpile after eradication? Because we have two choices. We could use Sabin's IPV, or we could use, Salk's IPV or Sabin's OPV. Now, Salk's IPV is made from virulent viruses. So in an era where polio is eradicated and no immunization is occurring, growing that virulent virus is a BSL-4 proposition. That's very hard to do production BSL-4. Sabin vaccines, of course, you don't need BSL-4, but they revert. If you had an outbreak, say, and gave Sabin vaccine, you'd now reintroduce all that vaccine-derived polio into the environment. So that's not a good idea either. So this, I think, is a very neat idea. And what was done here, this is in the lab of Eckhard Wimmer. They took polio, and they took the capsid region, and they changed every codon. So codon, of course, codes for an amino acid. They changed every codon so it would make the same amino acid, but the sequence was different. So you know the code has wobble, so you can do that. And they can make thousands of changes in this virus. It still grows, but it doesn't kill mice. It doesn't paralyze them. So it would, in theory, be a good vaccine. And if you have 1,000 or 2,000 changes, it's probably never going to revert. Because polio changes 1% a year, it's probably not enough time to get it to revert. So this would seem like the ideal vaccine. So you do this massive change in the capsid region. So this has to be clinical trialed, of course, and it's not clear who would do this. But this would be an ideal kind of vaccine to stockpile after eradication occurred. All right, I think I will stop there. Let me just move to one last slide that I want to promote my educational doings. So since reading Stephen Hawking's book, I've decided I want to be the world's virology professor. So I started podcasting and blogging. We now do 3 podcasts, one on viruses, one on bacteria, and one on parasites. And you can find them all at my blog, which is at virology.ws. I also write periodically about what's going on in the world of viruses. So check those out if you like viruses. Thank you.
Thank you very much. Please join me in thanking the speaker. I was going to mention, and thank you for showing that slide, if you want to keep up to date on what's going on in virology, this is a great source. And for those of you who are interested in learning about the scientific technical aspects, I highly recommend Principles of Virology, the book that Professor Racaniello co-authored, as an excellent scientific introduction to the field. So thank you very much. We'll have time for a few questions. The microphone is in place, and please feel free to ask.
Hi, thank you very much for an interesting talk. You mentioned earlier the rise of an anti-vaccine movement in this country. And what's your take on this today? Because we've had reactions in different contexts to the populace regarding as experimentation, the Tuskegee experiments or the atomic soldiers, et cetera. Where are we at today in our society with this?
So there's a growing anti-vaccine movement which stems from a number of sources. There have been some problems with vaccines, and one of them I pointed out here, the Cutter incident, began it all in the 50s. Other vaccines over the years have had side effects, and these I think, when amplified and used in the wrong hands, can be used to misinform people about the general safety of vaccines. So I think that, and couple that with the internet now, and that everyone can write whatever they like. There are many blogs which you can find which say this vaccine is bad because this happened and that happened, and it's all false, pretty much. And people find these and they think they're real, and that's in part why I'm doing this, to try and counter all of that. So there's an access to information, but it's not necessarily correct. And there's a perception that I don't have to immunize my kids, they'll be okay because there's not much virus around anyway. So measles, for example. There's not much measles in the US, so I'm not going to get my kid immunized because it may cause autism. And this is bad because if you have pockets of resistance, which you tend to do in communities, because anti-vax individuals tend to get together, they have meetings, and so you have in communities growing numbers of individuals who are vaccinated. Then you have susceptibility problems. Now there are outbreaks of measles in the U.S. My take is that this is highly unfortunate, of course, and it's increasing. And I'm not sure that anything I do or other scientists do has had any effect. I think the press is partly to blame for it. They still give credibility to Andrew Wakefield, first started this anti-MMR years ago. CNN recently had him on and said the controversy continues. There is no controversy. Why do they do this? So CNN wants to have people watching, so they make it controversy. So I have a lot to blame on the press for continuing this. They should say it's over, let's move on, but they don't. So that's the combination of misinformation, the internet, and bad press, which perpetuates. And I don't see it declining anytime soon. I think it's a problem. It scares me. I'm glad my kids are all immunized and on, to be honest.
Sir, thank you for the talk. It was fascinating. You said that only 1 in about 100 infections ends up with an infection of the CNS and then paralysis. Is there any information as to what causes that and whether it's with the virus or the host?
Yes, we do. And it's all from mouse experiments using the mice that I told you about. But let me tell you what it is. We think that there are two main things that cause the inefficient infection of the CNS. So when you first get infected with polio, it replicates in your gut and then gets into the blood, and it can get into many tissues. When it does so, it induces a very strong innate immune response, and that consists of interferons being produced. The interferons induce antiviral genes, and that limits polio to the periphery. So we think in 1 in 100 people, those individuals actually have a suboptimal innate response that allows the virus to get into the CNS. So if you take mice and knock out the innate system, which you can do genetically, then 100 out of 100 infections will get into the CNS.
Sir, would that lie with a general immunodeficiency, or is that an interaction with a different illness or disease, or is it just genetic?
So it's not a B-cell immunodeficiency. We don't have a lot of information on what kinds of mutations there are in the general population that would do this. So we're starting to learn for other infections that polymorphisms or mutations in these interferon genes do control susceptibility to virus infection. But there's not enough polio left to be able to do these correlative studies in people. So we don't know what kinds of mutations are leading that. The other is that the transport of virus from the muscle to the CNS, this is called axonal transport. It's a very specific system that we have to bring things from our muscles to the CNS and back. This is very slow and very inefficient. And that's probably another reason why the virus doesn't get to the CNS very efficiently. If you stick a person with a needle, say, let's say you give a kid a needle injection, and then the next day give them a polio vaccine. Those kids are more likely to get virus in the CNS. This has been known since the 50s. So if kids went to the physician and had a shot in one arm and a polio shot in the other, they would be more likely to get paralytic disease. The reason is when you stick a needle in a muscle, you are stimulating axonal transport because the muscle is responding to a stress and it needs to get things to the CNS and back. So that takes up the virus more quickly. So that's the other limiting factor, the slowness of the axonal transport. This is in mice, right? I don't know if it applies to humans.
Thank you very much. So earlier in your talk, you mentioned that one of the key reasons why polio is so deadly and has been so effective in its spread is because of this asymptomatic stage that varies in length, but is really crucial for the spread because you really don't know what infection someone has, and it's 1 to 1,000,000 that it could be polio and you really can't track it. So this asymptomatic stage has been observed in other viruses, and it's theorized to exist in some particularly infective ones. Ones that don't have it, like smallpox, for example, we heard about that. But influenza, for example, it's been not necessarily shown, but theorized that it does exist in that. So, given your experience with polio, do you think you could suggest any biological and also policy-related measures that we could apply in countries where this asymptomatic stage is particularly I guess, important for the spread of the virus? In other words, to combat this asymptomatic stage and any effects it might have.
It's a real problem because the way we do polio surveys right now is to look for lameness. Well, lameness surveys, which is looking for paralyzed limbs. And then when you go into a village and you see a lame child, you then get a stool sample and look for polio. But what about the ones that are infected but don't have paralysis? I don't know of any way. You can't sample everyone. My dream is that in 10 years we'll have a little scanner that you can run over someone and it'll tell you what viruses they have. I think of Star Trek, it was called Triplet. I am convinced that we will have such diagnostics at some point. In fact, I'm convinced in the morning we'll all stand in front of our mirror and it will read out all the viruses and bacteria infections we have and what to do about them. But that's obviously fanciful thinking, and it's not going to happen today. So at the moment, there's just no way to detect. You do sewage surveys, but they're only as good as the sewage you sample. And that's currently what is done to try and get an idea of how much virus there is in an area. But in areas with conflict, you simply can't do that. If you don't have a sewage system, you can't do it at all.
Hello, thank you for your talk. And in the pharmaceutical case, before that, there were no liabilities for pharmaceutical companies?
There were some, but it had not evolved to the point where the idea arose that if the pharmaceutical company made a product and it caused harm, no matter what was done, even if it was made perfectly. So in polio's case, this was made imperfectly, but even if you made it perfectly, and it's some inherent feature of the virus that causes it to paralyze 1.4 million cases, the company was still liable. So you can imagine all these paralytic cases that resulted from the Sabin vaccine. There were many, many lawsuits. Many were won because the idea is even if you made it perfectly, you still have to be liable.
So if today you get infected by something you took from Pfizer, they are liable always.
Okay. If you have a good lawyer,
okay.
The other side will have a good lawyer too. You start with the assumption that they are liable. Okay. So I have been on many of these cases as an expert witness, and there's no evidence that the vaccine is made improperly, yet most of the time the companies are found liable. And I think, I'm not a lawyer, but I think this began with the Cutter incident.
Thank you so much.
Sir, thank you. One quick question. I was wondering approximately how long does the virus stay active once it's in excrement in the waste process? So about how long will it stay floating around that we would actually have to keep on vaccinating for?
Well, it's a good question. The environment and the people are two different things, right? So in the environment, maybe at the most 6 months.
Okay, so the biggest threat is within...
Yeah, but these immunocompromised people who are shedding it constantly, they're not sick. So the virus is always growing in them and they're excreting it. So that is the problem. Because even if it lasted, 10 days in the environment, those individuals would still be
Okay. What was the longest case so far? I think I saw on the board that someone was estimated to have it for approximately 40 something.
Excretion in an immunocompromised individual, I think, is 15 years. So this woman in Minnesota, she had been 12 years excreting. So that's, to me, that's the real threat. We don't know how many of these people exist globally. It's very hard to diagnose them. And so, but we know that there are immunodeficient people, but whether they're infected with polio, we simply don't know. They're not screened. But in my view, that's the biggest problem. And it looks from all of these outbreaks of vaccine-associated disease that I talked about, they're all caused by virus from immunocompromised individuals.
Thank you.
Yeah.
Sir, one more question. With the goal of an eradication, how much of an issue do you think underreporting is? And I'm thinking in particular about areas where, like you mentioned earlier, there's an ongoing conflict, perhaps areas where medical care is denied to, for example, female children. And then that in combination with the fact that many Western or more and more Western personnel is in those countries, whether it be NGOs or Western armed forces, is it possible to stop immunizing as long as underreporting is going on? And is that an issue or not?
It's a huge issue, absolutely. I think you have underreporting of lameness, and also you don't detect virus because you don't look everywhere. So we thought type 2 was eradicated. Find it anywhere, but it was still there. It was in people. So I think both are major issues, and that's why even if you stop, if you say there's been no polio cases for 5 years, and you stop immunizing, you have to keep up the surveillance for quite a while. So this is going to go on for a number of years, a long time.
Will it be possible to ever stop immunizing?
Here it is. Whether the situation allows it to happen is really beyond WHO's rotaries control, right? Because it all depends on immunizing for a long time. So in theory, I think it could be done. I would like to see it done. I think it would be terrific, but it's really up in the air.
Thank you very much. Please join me in thanking Professor Racaniello.